So good morning, ladies and gentlemen, dear colleagues. May I kindly ask you to take your seats so that we can start this session. Good morning, ladies and gentlemen, dear colleagues. My name is Helmut Gaugic. I'm the head of Unit Land Use and Biosafety at the Environment Agency in Austria. And I would like to say that it is my great pleasure and also a big honor to welcome all of you here to this breakout session on advancing environmental risk assessment. I would like to welcome all the speakers and also, of course, all of you here in this audience for this very important session, which is organized by the European Food Safety Authority. Together with my colleague, Jock Martin, who is heading the Integrated Environmental Assessments Unit of the European Environment Agency, I will have the pleasure to chair this session. And I'm really looking forward to it. I think it is a very important and very interesting topic which we have in front of us. And I'm very much looking forward to good presentations, to a good debate, and to a good outcome by 1 o'clock today. Just a few words of introduction. What is the topic of this session? As most, if not all of you know, regulated products such as genetically modified organisms, but also such as plant protection products, food and feed additives, they have to undergo an environmental risk assessment before they are being placed on the market. And they are subject to an approval process before entering the EU market. The European Food Safety Authority has a very important role in this process, and that is to provide scientific advice to the risk managers on any possible risks these products may have on the environment. Now, environmental risk assessment for that purpose is a quite well-established process and mechanism, but of course environmental risk assessment also still poses quite a lot of challenges and opportunities. And the objective of this session, in fact, is to discuss some of these challenges and opportunities of environmental risk assessment and to discuss on how we can advance environmental risk assessment. It would be quite a broad topic to discuss all the challenges. We, EFSA, decided to pick 3 important ones. And these 3 ones which we are dealing with this morning will be on harmonizing approaches to making protection goals operational in the environmental risk assessment process, first topic. Second topic will be paying greater attention for the relevance and reliability of scientific studies and data supporting environmental risk assessments. And thirdly, quite a big challenge, moving towards integrated environmental risk assessments by including multiple stressors, approaches for including multiple stressors. So these will be the 3 main topics of this morning's session. We will discuss these 3 points also one after the other, and then have a moderated panel discussion taking all these 3 topics together and allowing you also to bring in your viewpoints to this process. Now, just a few organizational points at the end, in order also not to be too long at the outset. I would like to mention, as it has been yesterday and the day before, that this session is also live broadcasted. And I would kindly ask participants to identify yourself before you take the floor when asking for an intervention from the audience. Just mention your name and your institution. Questions with relevance to this session can also be tweeted during the entire session. Here the tweet address can be seen. We are looking forward to inputs from the audience, but also from those who are following this session via live broadcast. We have a slight change in the program. Our colleague Steve Ratton from New Zealand was unfortunately not in a position to come due to health issues, but I'm very glad that Glenn Suter from the US EPA was offering to jump in the very last minute. I would like to thank you very much, Glenn, for kindly offering this possibility on such a short notice. Colleagues, please also switch off your mobile phones during the debate. And that is all what I wanted to say in the beginning. And we are now moving to the first presentation. I'm very pleased to invite Professor Alan Gray from the Center for Ecology and Hydrology to come to the podium and to give his keynote presentation. We are glad that Alan is here. He was, until recently, still struggling with some virus, but obviously was managing to cope with it just in time. So we are glad that you are here. Alan is a honorary research fellow with the Center of Ecology and Hydrology in the UK, where he has retired in 2003, but since then still very, very active. He's currently president of the International Society for Biosafety Research, and he has been involved with GMO and biosafety in the UK since the 1990s, among other things, being member and chairman of the Advisory Committee on Releases to the Environment. Alan, you have the floor, and we are looking forward to your keynote presentation. 20 minutes. Thank you.
Thank you very much. I hate the word keynote. It sounds very grand, which this talk isn't. What this talk is to introduce basically what's going to happen during the rest of the session and to give a personal perspective on environmental risk assessment. I guess, well, I don't have to guess because I can see in the room many friends and colleagues who, like me, sort of stumbled into environmental risk assessment, were innocently doing their scientific research, in my case, on genetic variation in natural populations of plants in relation to their breeding systems and gene flow and for conservation biology, but were pulled in to give advice about this new phenomenon which we encountered in the late 1980s, early 1990s, of recombinant DNA technology producing plants with genes they wouldn't normally have. And there's the usual disclaimer because it is very much a personal trip. What I want to do really is to take you back to those days when we gathered in rooms, virologists, ecologists, molecular biologists, and soil chemists and so on, and scratched our heads about what might happen when we grew these crops. And secondly, to look at some of the changes that have gone on during this process of since the early 1990s in our thinking about how to do environmental risk assessment. And finally, just a couple of what I believe to be the most interesting of the many challenges, of which there are a lot, but these interest me. So this roughly is a definition we can use. I'm dealing principally with GM crops, but of course others will cover other aspects of environmental risk, I'm sure. That will do as a working definition, but just to remind you principally why we do it so people can make decisions about activities like growing crops or using agrochemicals or whatever. And it's only part of a process. It isn't the whole thing, and risk management and risk mitigation is a key aspect of those three things to do with risk analysis: risk assessment, communication, and management. And we can do these sorts of ERAs at all levels, for putting the crop on the market for commercial release, for having a trial like this one of cassava in Uganda, and for importing live seed for feed, food, or processing. And they will have different contexts if we do different risk assessments. Just a reminder of the timeline in all this. I mean, it was very clear early on in the mid-1970s that this technology had the potential, if not the actuality, for doing some really interesting things, but things that we ought to have some oversight. We should have some regulatory oversight. We regulate these things not because of what they actually have done, but what they could potentially do. And in the 1980s, some of these frameworks were being developed using existing regulations, and the first commercial production of the GM crops saw very similar, basically similar risk assessments in different countries, but using different legislative frameworks. In the USA and Canada, for example, they took advantage of existing regulations where Australia has their Gene Technology Act, and we in Europe have a series of directives which cover the release of GM crops. But mostly they were treated as different, as fundamentally different from non-GM crops. And through the 1990s and beyond, a series of international agreements sort of set the broad framework for our regulatory environment, the broad principles of environmental risk. When we first sat down in these rooms to think about what could happen, we came up, I think, with a what could go wrong stage, with this great infinity of possible things that could go wrong. And we were accused of having a lack of imagination. And even when we went to the known unknowns, we were urged by many to try and think of things that might not happen, the famous unknown unknowns that we couldn't get at. And I don't know why that was. I mean, I think partly because somehow it seemed a bit scary to have genes out there instead of chemicals, the genie out of the bottle concept. And there's also the distrust of what seems to be a simple approach, but I'm sure you'll hear it isn't, of the agrochemical pesticide approach. And also ecologists love complexity. We like the old butterfly's wings stories. We like something to be multivariate and so on. We love models. We believe the world is a complex environment. So maybe we thought simple ways weren't appropriate. So what happened? I mean, this is Mick Crawley coming up with a list of the harms that could happen. I don't know how far the story of chicken licking has spread from my nursery rhymes, but the sky was about to fall in, I think, for most people at the thought of releasing these crops. And these were some of the things which British ecologists came up with that might happen on the growth of the crops. One of the trends over the years, of course, has been a more tractable set of harms. I mean, these are just the five that the Canadian regulators think about. They regulate in a different way because they do genuinely talk about the product, not the process. So any plant with a novel trait, however introduced, is subject to a risk assessment. And these sorts of things, you're going to hear a bit more about them, becoming a weed, through gene flow to wild relatives, becoming a weed or invasive. Plant pest is peculiarly Canadian, and the impact on non-target species, you're going to hear from Jorg, and the potential impact on biodiversity, which I'll come back to. It's a sort of catch-all phrase. In Europe, there's a rather longer list, and it includes some interesting harms, which may be more difficult to evaluate, but certainly have a socio-economic element, like cultivation and harvesting and the effect on production systems. A lot to do with what farmers decide to do, what the take-up is, and so on. So that's the list of harms. And some of those in the early days we thought were possible, I think we've sort of put into the negligible area. Things like the emergence of novel viruses from viral recombination, Mark Tepfer's recent paper and his colleagues covering that. The realization that actually there's huge variation in microbial populations in the soil, and that functionality is the important thing to think about there. And perhaps from my own area, and controversially, the idea that natural habitats would be invaded by GM plants or hybrids between GMs and wild relatives. I'm happy to argue that with you later. Ecologists love models, and one of the early models was, will these plants behave like weeds or invasive species? So some of our weeds from the UK here, the giant hogweed and Japanese knotweed, our famous weeds, would there be parallels there? And I think very quickly that was rejected really in favour of more of a sort of crop model. It has very poor predictive power. If you add weedy characters to plants, they don't necessarily become more weedy. We know what the weedy characters are, and we've found that in weed risk assessment, things like dormancy and rapid early growth rate and so on are good things for us to think about in weediness, but actually you get very poor predictive power, and more like a quadratic here when you look at the sorts of weed characters, so-called Baker traits, added to various plants in relation to their weediness. So what have been the conceptual changes over these years since the mid-1990s or the early 1990s? One of the things that I think's happened, and this is from an interview I gave to a newspaper, and they rather liked the idea, is that we've gone from talking in general about GMOs and GM crops, the sort of broad ideas and generalizations, to rather more specific detail, to objective, analytical. We've gone from sort of Platonic to Aristotelian. And people used to talk about, GMO in this general sense. I was often asked by the ministers I advised, are you in favor of GMOs or against GMOs? And I would try and say, well, actually, that's not a legitimate question, really, Minister. They are. It's a technology. It's like asking me, am I in favor of vaccination, or am I in favor of molecular, using microscopes or whatever? So we've gone more and more from this very generic to a rather specific trait-based approach. So we think specifically about BT crops, about herbicide tolerance, about virus resistance, and so on. And we have gone more, I think, from the process to the product. Effectively, we are doing what the Canadians do and looking at plants with novel traits. Not universally, but we have gone from looking at plasmid maps and insertion sites and things like that to assessing the phenotype and its impact. So I think that's one thing. But the most important thing that's happened, certainly in Europe, but was happening elsewhere earlier, is to use problem formulation as the first step in the environmental risk assessment. Now, a huge amount's been written about this and whether there are 4 steps or 5 steps or whatever, and you're going to hear some more from people who are very used to using this particular technology. But my definition is that what it does, it puts the ERA in a way which identifies what it is you're trying to protect and asks what harm's going to occur to it by the cultivation of the crop, and then asks you what information you need to assess the likelihood and seriousness of the harm occurring. So, one of my ways of looking at it, which is what I think we did in the early days without actually explicitly knowing we were doing it, is asking what do we want to see protected? Can we envision a way in which it might be harmed? And how can we assess, how can we link the growing of the crop to the harm that we're anxious about? And does it matter in the regulatory context? So the key features of problem formulation, and we're going to hear more from people who in later talks, are that they develop these operational protection goals from broad policy protection goals. I'm going to go into this in more detail. And they set the context of the risk assessment, depending on where it is, what it is, the crop, the receiving environment, and so on. And because it seeks to test specific risk hypotheses rather than do what we scientists normally do, which is test null hypotheses, and focus on the data that we need to test the risk hypotheses, it avoids us what's called a deficit model, the idea that the more we know, the better will be our risk assessment. If we just keep on adding information and understanding to the situation, we'll make a better risk assessment. And that simply isn't true, as John Lawton showed with the deficit model. It decides the science that we need to know versus the science that we would like to know, that's interesting, and I think we're going to hear from Joe Smith something about that. And what it does is also promote a sort of what Alan Raybould has brilliantly called an ecotoxicological versus an ecological approach. This generally broad model building, rather weak predictive thing that we ecologists do as opposed to a more ecotoxicological approach. So those, I think, are the key features that problem formulation has brought. Let me finish with some current challenges. There are lots. I mean, I could have written more. I could have put the challenge of doing ERAs in low-resource countries, which is something which I personally get involved with a lot. But the 3 that I really want to look at in yellow there are this business of translating these broad policy protection goals into something that you can actually test that isn't vague, and agreeing what criteria we have for harm. I'd say a tiny bit about conflicts, but not multiple stresses, since we have a talk later on. And I'll just say a little bit about making the risk assessment more contextual, because I do think this is important, of looking at ways in which we can bring a wider purview to the whole business of assessing risk and harm. If we look at something like this policy protection goal, this is a pretty famous one in the Cartagena Protocol, we can see that there are words in there like potential adverse effects, harm, conservation, sustainability, and biological diversity, which are really challenging to a scientist. And we're going to talk a little bit more about this during the session. And getting those into something we can measure and turning into assessment endpoints is really one of our biggest challenges. One way we might do it, and I think Professor Mob is going to talk about this, is we can think about the function, the ecosystem functions or the ecosystem services approach would help us to define some of these protection goals. If we're interested in crop pollination, then clearly populations of insect pollinators will be something we can measure and be concerned about and monitor. If we're interested in sustainable agricultural production, in-field weeds, again, would be something that would be a way of measuring whether there is a harmful impact or not. And what is often not realized is that, of course, different habitats in our European heterogeneous farm landscapes are very different protection goals. So the protection goal of the sugar beet field is to produce sugar beet sustainably and over a long period of time. But the goal of the semi-natural habitat on the right, the chalk grassland, is the maintenance of a highly biodiverse communities of plants and animals. And of course, occasionally, as in the UK, I think I put the EU there, but it's true of the EU too, in the Northern Europe, different protection goals may lead to conflict where in fact they relate to the same environment. And perhaps the classic for us in England has been farmland birds and weed-free crops. One wants crops to be produced in the fields, but at the same time there is a possibility that making all of our fields weed-free would make a huge difference to those birds that are much loved by the British, as you know, that are dependent on that habitat for part of their survivorship. And here's an interesting case where risk mitigation could be used, but again, we could go into that in questioning. Interestingly, the model of this, the most unknown variable was farmer uptake. It wasn't a bit of scientific understanding, it was a bit of socio-economic behavior, which is fascinating. Conflicts also arise in the non-agricultural environment, which is something people think, is it the natural versus the unfarmed environment? Of course it isn't. This is one of my little favourites. These lovely creatures, hedgehogs, were introduced into a Scottish island in 1974 to kill garden pests. They spread and have become significant predators of the eggs of rare and much-loved nesting shorebirds. So here's a situation where we know the science, we know what's happening. What needs to be done is agreement about what we do with the hedgehogs, whether we kill them or cull them or remove them or trap them or whatever. Trying to do more research can probably, as Sarovich showed, make the situation worse. Conflicts do occur in conservation all the time. I've taken this little quote from Jan Devos's paper. We can't protect everything all the time, and we need to pull the values out of what is going on and explicitly discuss them and debate them openly. I happen to be involved with this series of books published by the British Ecological Society. And this one, I recommend this one in Conflicts in Conservation to you. It shows really, there's many, many examples of how you work towards solutions when there are clear conflicts in the natural environment. Let me just finally finish by saying what I mean by making ERAs more contextual. This rather horrifying picture is based on what I said when I was appointed as chairman of ACRE in 1999. So we're going back into the Stone Age here, folks. And what I said basically was that compared with what we've been doing in our countryside, speaking as an ecologist, conservationist, without totally unregulated, what we were likely to do with GM crops, highly regulated, faded into insignificance. Unless we take notice of things, changing baselines and changing agriculture, we're never really going to put the risk of harm into context with the benefit. We need to insert into the whole picture of agricultural change the idea that we are in any way changing our countryside. I'm not actually in the picture there on this picture nearest me, but I am old enough to have done this. I have worked with horses on a farm in rural Lincolnshire in my father's farm, making hay by throwing it up on a thing. And over in my lifetime, this huge change has happened, where we've gone from haystacks to bales to silage wrapped in plastic, and from old pasture full of wildflowers to temporary lays of ryegrass and clover. And unless we uncouple the idea that GM crops lead to modern industrialization, de facto they have in some countries, in Argentina and so on, but it is not necessarily true, and that we may be losing benefit by not taking into account some of these aspects of the use of modern biotechnology in our landscape within Europe. So, and I think this is particularly true in the wider context where you think about what actually happens in non-GM agriculture in the tropics. But I've seen in 2 or 3 papers, the paper which Acre put together, an EFSA paper, and again something which the very thoughtful Jan and his colleagues put together, these words are paradigm shift. It would require, I think, in the way the EU currently do environmental risk assessment for GM crops, a paradigm shift to bring into account, to pull into the broader picture, the sort of harm and risk assessment, the assessment of harm. So thanks for your attention.
Thank you very much, Alan, for an excellent keynote presentation. Thank you for sticking exactly to the time. That's great. That's very important, also because this session is live broadcasted. I'm really appreciating that. Now, as you have been sticking to the time, there is the possibility for about 1 or 2 questions. I would kindly ask you maybe at that point in time really to restrict it to questions of clarification, not comments or discussion, because we will have a moderated panel discussion at the end. And please identify yourself by mentioning name and institution. 1, and then over there, please.
Yeah, my name is Detlef Bartsch. I'm from the Federal Office of Consumer Protection and Food Safety, and I came a long way along with you, Alan, in the early days, and I'm happy that you started with this historical overview. My question is about the definition of harm, because this is key. And in the definition of the GMO law, we have this in many legislation, a definition that GMOs are unnatural. So it's kind of alien species out there. Has there been a mistake made in these days? Because many people think that a GMO in the environment per se is harm is unacceptable, and then we can stop any risk assessment, because if we want to bring them out, they are there.
Thank you, Detlef. Alan?
Thanks, Detlef. Yes, I'm aware that that view is around. I think the scientific consensus about the harm that these crops can do wouldn't support that in any sense. So I mean, I think we do need to continue to think about how modern biotechnology might both improve yields and bring what we need in terms of advantages to agriculture without harming the countryside and other places around. So I think that's a sort of fundamental threshold that people have. If it's your belief that inserting genes in this way is unnatural or difficult, I mean, the longer we go on, I'm a co-author on the Ladix paper, where we look very carefully at comparing the amount of unintended molecular and genetic effects that occur in recombinant DNA technology compared to conventional plant breeding. I think we have arrived at the stage where we do understand that this is on the same sort or even lower level. They are not to be treated from the scientific point of view, and those of us who deal with the phenotype most of the time, in some way as though they are alien. I find that a difficult concept, but I do know it exists, and I do understand why it exists.
Okay, thank you very much. Time for one more question. The gentleman over there, please.
Thank you for the brilliant presentation. I would ask one question. Don't you think that in addition to the two models you propose, especially for developing country, one is GMO-based, the other heavily chemical-based model, there are also a third way that is, let's say, the agroecological models that in any case are able to feed, to produce 70% of the food for the planet.
Thank you. Alan?
Yes, I mean, I think that is true. Where these crops have had most impact, of course, has been particularly in developing countries and in areas like sub-Saharan Africa and India and Pakistan, where, particularly in Africa, where farmers who are mainly women lose 60 to 80% of their crops often in the field and through storage, and where protection against insect pests not only increases the yield and enables them to save food and to sell food and send their children to school, but has really fundamental effects compared to the way in which we would use these in Europe. I mean, I fully accept that in Europe we have a different problem. We heard something about that in the sessions yesterday to do with obesity rather than food security. And I was reading in the Times the day before yesterday, we've had record wheat and oilseed rape yields in England. We've beaten New Zealand apparently in the yield per field or per acre for wheat this year. So we're moving in that direction without these sorts of things. But there are places where I agree that it would be important to think about some technologies which could help to fight pest and disease.
Okay, thank you very much. Thank you once again, Alan, for an excellent keynote presentation. Thanks for your questions at this stage.